I am Alberto Paradela from the Functional Proteomics Laboratory of the National Center for Biotechnology, Madrid, Spain. In this presentation, I will show you a summary of the results that we have obtained in several tests that have been performed with the automated sample preparation platform AccelerOme. Our objective has been to evaluate the efficiency of this platform in the digestion and labeling of complex samples with the TMT isobaric labeling reagent, assessing if it is possible to integrate the AccelerOme platform in the routine workflow of a proteomics laboratory. The platform also allows the triptych digestion of samples in the absence of TMT labeling for the subsequent performance of label-free quantitative proteomic experiments, either in data-dependent or data-independent formats. Results for this kind of experiments will not be shown in this presentation. The AccelerOme automated sample preparation platform has this external appearance with relatively small dimensions, a volume of about half a cubic meter and a weight of 70 kilograms. It can therefore be easily placed in any laboratory without any relevant space problems. It has two compartments. On one side, the first one for the bottles containing the mobile phases, aqueous and organic, and the second for a hand controller to set several technical parameters of the instrument. The platform is also equipped with a QR code reader for the QR codes displayed in the TMT labeling kits. There is also an integrated touchscreen display with user interface for instrument control and operation through a graphical wizard. The inner space of the equipment is accessible through a glass door where we can find market space for the placement of the various elements of the kits, as well as the robotic arm for liquid handling. The platform also includes an integrated miniaturized spectrophotometer to measure the final peptide concentration of each sample. Inside the AccelerOme platform, there are different spaces clearly marked to place the elements available in the different TMT labeling kits. First, we have the space where the sample input plate is located. This space allows the samples to be heated and shaken. On the left, in green, the standard arrangement of the samples in a two-experiment labeling reaction with TMT 16-plex. In the position B, there are two clearly marked spaces. First, B1 for the location of the rack with the kit reagents, proteases, peptide standard, enzyme, reconstitution buffers, and TMT. And B2 for the rack with the TMT reagents, TMT 16-plex in this case. Several notches located here and here avoid the possible errors that can be made when placing the racks. Next, we have the space to place the sample washing and elution solutions used to clean the samples once they are labeled and mixed. Micro solid phase extraction cartridges that are used for this purpose are placed in this position. Finally, the output plate where the sample are collected once the process is finished is located here. The different TMT reagent labeling kits available for use with the AccelerOme platform allow simultaneous labeling of one or three reactions in the case of TMT11, or one or two reactions in the case of TMT16. The amounts of samples that can be labeled are also variable and range from 10 to 30 micrograms in some cases, and 30 to 100 micrograms in the second case. Kits for sample digestion and label-free quantitative analysis are also available in two versions, allowing for simultaneous digestion of 16 to 36 samples. Here I summarize graphically the different experimental steps necessary for a successful result, the initial steps for the preparation of the samples that will be later be processed by the AccelerOme platform are performed externally and are described below. The scheme summarizes the recommended procedure in case the lysis buffer that is included in the kit is used, and ends with the quantification of the samples by standard colorimetric analysis. We have not evaluated the use of alternative sample preparation procedures in our tests, although we anticipate that they will be feasible, provided that they do not interfere with the subsequent digestion and labeling of the samples. Once the samples have been processed and quantified as described in the previous slide, they are placed on the input plate. This figure shows what will be the arrangement of the samples in an experiment with a total of 32 samples corresponding to 2 independent labeling reactions. Obviously, always will contain an identical amount of samples, ranging from a minimal value of 10 micrograms to a maximum value of 100 micrograms, depending on sample availability and the type of kit that we have decided to use. The final volume in each well should not exceed 50 microliters. Once the equipment is turned on, select the Quick Start Setup option and then scan the QR code of the kit to be used with the help of the handheld QR code reader. Finally, we will proceed to place the different components of the kit. First, fill bottles 1 and 2, the first one with water plus 0.1% of formic acid and the second one with a mixture of water and methanol 50-50% and formic acid as well. With at least, the bottle should have at least 250 milliliters. Next, we will proceed to place in position D the rack containing the bottles with the washing and elution solution. In the same way, we will proceed to place in positions B1 and B2 the digestion modules and the TMT reagents, respectively. Next, and sequentially, we will place the rack with the solid phase extraction microcones and the output and input plates in their respective positions. We will confirm again that all the components are in their corresponding locations and that there is no elements that could interfere with the correct operation of the instrument. Finally, we will proceed to start the protocol. It is important to note that from the moment that we select the Quick Start setup options and scan the kit QR code, the touch screen will sequentially, as shown here, indicate each of the steps mentioned above, inviting us to move on the next step once the previous one has been complete. Finally, we will proceed to start the experimental sequence. The screen at that moment will display an estimate of the time needed to complete the entire procedure, typically between 5 hours and 5 hours and a half. There is an option to abort the process that can be useful in case we observe that something is going wrong and we can still rescue the samples. The graph below shows the time sequence of experimental step performed during a typical TMT labeling experiment. First, each of the TMT tags is dissolved with the TMT reconstitution buffer. This is followed by reduction and alkylation of the samples on the input plate and subsequent digestion with lys-C and trypsin. After labeling, samples are mixed, cleaned in the solid phase SPM microcolumns with the wash and elution solutions, and then placed on the output plate. The final step consists of measuring the peptide concentration of the labeled samples. At an intermediate point of the above process, the instrument has obtained the calibration curve that is necessary for the calculation of the peptide concentration. Once the reaction is finished, the system will generate a report in which we can save, first of all, a summary with the data corresponding to the method and kits used, the lot number, the location of the different samples on the input plate, and the location of the reaction product on the output plate. Here it's important to note that each labeling reaction generates two subsamples, which are in principle equal. Secondly, we have a table and a graph with the values obtained in the calibration curve with the peptide standard. Thirdly, the calculation of the amount of labeled peptide obtained in each reaction considering the values of the calibration curve. And finally, and according to these values, to the values of the calibration curve, and according to these values, and the final concentration values that are set in the instrument, in the system, that can be changed, it has to be set, a series of recommended values to reconstitute the sample to the desired concentration. Finally, a table summarizes the position of each sample on the input plate, which TMT tag was used in the labeling of each sample, and the position of the sample once mixed on the output plate. Next, I will show you some results that we have obtained in our laboratory in some tests performed with complex samples. First, we used a test sample formed by the combination in known proportions of 3 proteomes, extracted from HeLa, Escherichia coli, and Saccharomyces cerevisiae, respectively. The proportions or amounts that we use in this design experiment allowed us to design 4 different conditions known A, B, C, and D, with ratios ranging from 4 to 1.3 in the case of Escherichia coli and from 0.66 to 0.33 in the case of Saccharomyces cerevisiae. In some cases, the ratios corresponded to the absence-presence values, as here, while in the case of HeLa human proteins, the ratios should remain around the value 1. We selected for the first test the TMT 16-plex kit for 2 reactions of 16 samples each, for 10 to 30 micrograms of sample. For this experiment, we labeled 25 micrograms per sample. For biological replicates, that is, samples that were independently prepared, were labeled for each of the 4 conditions A to D. The labeling schema is as shown in the figure. This labeling schema, experimental schema, was repeated once more, 1 week apart, in order to test the reproducibility and robustness of the AccelerOme platform. You see here that there is a 1 week difference between both experiments. 3 technical replicates of each samples were analyzed. The comparison between the sample quantities calculated using the values obtained in the integrated microspectrophotometer shown here and the values obtained in the colorimetric test before the experiment, that is when we prepare the samples, shown here, suggested recovery yields around 65 or between 65 and 80%. However, it is important to say, or we must take into account, that both the initial sample values, input, and those obtained with the instrument, with the AccelerOme platform, the output, were obtained using different techniques. According to these values and the parameters set in the equipment, the AccelerOme platform calculated a sample reconstitution volume suitable for injecting into our LCMS platform a total amount of 1000 nanograms or 1 microgram if you prefer, in a final volume of 5 microliters. The analysis was performed on a liquid chromatography MS platform formed by an ultimate 3000 RS-LC liquid nanochromatographer coupled to a thermo Orbitrap Exploris 240 mass spectrometer. For each injection, 1 microgram of sample was analyzed with a flow rate of 250 nanoliters per minute and a total gradient length of 120 minutes, including equilibration times. The effective gradient length was 90 minutes. The figure in the upper panel shows the characteristic chromatographic profile of one of the replicates, where in addition to the correct separation of the peptides, the presence of a very prominent chromatographic peak is quite evident. This peak elutes for about 4 to 5 minutes and, as displayed in the lower panel, corresponds to a very prominent, to a major molecular species with a mass to charge value around 3078. The signal is really very prominent and could interfere with the isolation and fragmentation of other coeluting molecular species, mostly labeled peptides. But we have found, and this I cannot show this data, that it disappears if the sample is prefractionated by reverse phase microcolumns under basic pH conditions. We believe that this prominent contaminant at 378 could correspond to the presence of TEAV in the sample, which is not recommended according to recently published work. However, it remains to be determined whether this is the case or not. We first evaluated for this experiment whether the AccelerOme platform could perform an efficient tryptase digestion of the samples. To this end, MS1 and MS2 spectra obtained in each of the experiments, performed 1 week apart, we used to launch a dynamic search with a mascot search engine against a combined database consisting of the proteomes of Homo sapiens, Escherichia coli, and Saccharomyces cerevisiae. The results in the table showed very similar results both at the level of proteins identified, peptide spectrum matches, and identified non-redundant peptides, and a very high digestion efficiency. between 98 and 99% in all cases. We identify only between 1 and 2% of peptides with a single miscleavages, and the percentages of peptides with 2 miscleavages was very low. Next, our interest focused on determining the efficiency of labeling with the different versions of the TMT reagent. The dynamic search shows that the percentage of labeled peptides range from 93% to close to 100%. However, for each of the 2 experiments performed, the first block of samples was labeled with a higher, as seen in this case, with a higher efficiency than the second block of samples, here and here. The cause of the observed differences in labeling efficiency is not due to the TMT reagents used, since they are exactly the same in the 2 sample blocks. Our hypothesis, pending confirmation, is that these differences between the block 1 and block 2 of samples could be due to the appearance of some bubbles in the tubes as the experiment progresses, and would mainly affect to the second block of samples. Having demonstrated that the AccelerOme platform is capable of digesting and labeling the samples with high efficiency and processivity, we perform a quantitative proteomic analysis of the samples to determine whether the experimental ratios match the expected ratios. Quantitative data analysis was performed using Proteome Discover 2.5 software for identification, for the extraction of signals from the different tags, and for statistical analysis. A combination of 4 search engines was used for identification. This approach allows us to increase the number of non-redundant peptides identified by approximately 20%. As can be seen in the table, approximately 70% of the identified proteins correspond to human proteins, HeLa cells, while the remaining 30% of the proteins were almost equally divided between Escherichia coli and Saccharomyces cerevisiae proteins. The bulk of identified proteins could, about 97% of the proteins could also be quantified. Regarding the quantitative data, and focusing first on human proteins from HeLa cell, we did not expect differential proteins with statistical significance between any of the conditions, as the amount of proteins were exactly the same in the 4A to D conditions. According to the statistical analysis, and as expected, most of the human proteins did not show regulation, either positive or negative. As can be seen here, please note that the human proteins are represented in gray. Only a small percentage of the human proteins, between 1 and 3%, did show regulation with a statistical significance. These proteins can be considered as false positives, and the probable explanation for their occurrence is due to the minimal but real presence of shared peptides between the 3 proteomes. In the case of the quantification performed on Escherichia coli proteins, the expected ratios range from 4 in the most extreme case to 1.33. We see that a significant percentage of the approximately 550 proteins identified, ranging from 70% in the most favorable cases to 30% in the less favorable cases, showed statistically significant regulation values in the expected direction. Two extreme examples are shown in the graphs below, illustrating how the population of Escherichia coli proteins, here shown in orange, here and here, shows values more or less distant from the axis, that representing the absence of regulation. Finally, for the quantification performed on Saccharomyces cerevisiae proteins, the expected ratios range from 0.33 to not expression for condition T. A significant percentage of the 450 proteins identified, ranging from 84% in the most favorable cases to 54% in the less favorable cases, show regulation values with statistical significance in the expected direction. Two extreme examples are displayed in the graph below, illustrating how the Saccharomyces cerevisiae protein population, here shown in green, shows values more or less distant from the axis representing absence of regulation. It is important to note that in the setup of the proteome discovery software analysis softwares, the absence presence values appear numerically as 100 or 0.01. Overall, our data obtained from the analysis of this standard sample suggests that the AccelerOme automated sample preparation platform is capable of processing, digesting, and labeling with the TMT reagent with high efficiency and processivity. Furthermore, data analysis of the results indicated that these reflect the expected values. These results encourage us to perform further tests on complex proteomes in more defined biological models. For this second assay, we designed an assay using 3 different strains of Listeria monocytogenes and one of Listeria valentina, with the goal of comparing the variation at the proteome level resulting from growing the different strains in H or L medium. 4 biological replicates were grown per conditions. The kit used was the TMT Pro 16-plex MS sample preparation kit for 2 reactions of 16 samples each, designed for labeling between 30 and 100 micrograms per sample, unlike the previous example. The amount of digested and labeled sample in this experiment, was 30 micrograms. All other parameters of the liquid LCMS analysis were very similar to those used in the previous example. Experiment 1 was reserved for the labeling of 8 biological replicators, 4 grown on H medium and 4 grown on L medium, of each of the EGDE and CC1 strains of Listeria monocytogenes, according to the labeling schema shown in the figure. Experiment 2 was reserved for the labeling of Listeria monocytogenes strain CC121 and Listeria valentina strain Lisval 98, also grown on H or yield medium, according to the labeling schema shown in this figure. Total input protein per experiment was 480 micrograms, resulting for multiplying 30 micrograms per sample by 16 samples, according to the colorimetric quantification previously performed. The amounts obtained after digestion and labeling with the TMT reagents in the AccelerOme platform were around 400 micrograms and 331 micrograms, representing a recovery yield of 84% and 69% for experiments 1 and 2 respectively. The labeled and digested samples were resuspended at a concentration of 0.2 microliters in the volume suggested by the platform. Here, a total of 1000 nanograms or 1 microgram was analyzed by LCMS using a gradient of 120 minutes, of which 90 minutes were an effective gradient, according to the conditions mentioned before. We decided to independently analyze the two output samples of each experiment, named A1 and B1 and A2 and B2, as well as a pool of them. The chromatographic profile obtained for sample A1 from experiment 1 is displayed in the upper panel here, where it is remarkable that the huge signal that was detected at very initial times in the previous test and appearing here around this area and corresponding to a very prominent signal of 378 mass to charge ratio is not so prominent in this case, although it is still detected. The MS1 and MS2 spectra were used to launch a dynamic search against a combined database containing the reference proteomes of the different strains analyzed. According to the data, a similar number of proteins were identified in each experiment, corresponding to approximately 80% of the total proteome of each organism. The number of non-redundant peptides identified ranged from around 17,000 to 19,000 peptides, of which 95% and 97% for experiment 1 and 2 respectively corresponded to peptides without miscleavages, and 4.5 and 3% of the peptides to peptides with a single miscleavages. The percentage of peptides with 2 miscleavages was very low. Regarding to the labeling efficiency, the same combined analysis showed that it was close to 100% for experiment 1 and 98%, more than 98% for experimental 2. Taken together, these results demonstrate that both tryptic digestion and TMT labeling were highly efficient for both experiment 1 and experiment 2. Next, we perform an individualized analysis of each of the output samples separately against their specific database. The analysis showed a very similar number of protein groups identified in each of the output samples, A, B, and pool. We also did not detect significant difference, less than 5 to 10% in all cases, in the total number of proteins identified between the different Listeria monocytogenes strains, which indicates that the AccelerOme platform does not introduce significant biases in this aspect, in this point. Venn diagrams performed for each of the individualized analyses show a high degree of convergence between the identified proteins, with the percentage of specific proteins in each of the output samples or in the pool of samples being less than 2% for the EGDE and CC1 strains of Listeria monocytogenes. And as for strains CC121 of Listeria monocytogenes and Lisval 98 of Listeria valentina as well. However, the same Venn analysis performed at the level of unique peptides showed us a more complex picture. Thus, I'm focusing on the two output samples, A and B, produced by the AccelerOme platform per experiment. We see that approximately 25% of the total unique peptides identified are specific to either A1 or B1 output samples. A similar situation is reproduced for the A2 and B2 output samples, although here the percentage of unique peptides specific to either A2 or B2 rises to 30%. We have not explored the reasons for this difference, but we think that a possible reason could be that the mixing process of the samples, once elute from the micro-solid phase extraction columns, is not 100% efficient. If this were the case, the output sample would not be identical, which might explain the existing differences. Turning to quantitative results, we notice that practically 100% of the identified proteins could also be quantified, as expected considering the nearly 100% efficient labeling described in previous slides. We observe that the exposure of the different strains to L or H culture media cause variation in protein expression, either upregulation or downregulation, that affected approximately to 65 to 70% of the proteins identified and quantified. This is therefore a massive rearrangement of the proteome caused by the exposure to H or L culture media, as can also be seen in the volcano plot on this slide. Finally, the PCR, the principal component analysis, confirms that what we already displayed in a previous slide, that is, that there are significant differences between the output A and B samples generated from each experiment. Thus, we see for the labeling of the EGDE strain samples that the PCA analysis is able to differentiate not only between L and H populations, as here, but also to distinguish between the A1 and B1 populations, as expected, but also, as not expected, of each culture medium, leaving the pool sample in an intermediate situation. A similar situation is obtained in the principal component analysis of the samples corresponding to strain CC121, although in this case the pool sample is not located in an intermediate position between the A and B populations. Overall, the results shown here through this presentation demonstrate that the automated AccelerOme platform is capable of digesting and labeling with a TMT reagent, with a high degree of efficiency and processability in the range of 10 to 100 micrograms. The highly automated and attended process takes just over 5 hours and generated a quantified clean sample ready for subsequent LC-MS analysis. Our data also indicates that the output samples obtained after the experiment reflect the protein amount variation present in the samples. Therefore, we believe that the automated AccelerOme platform can be a useful and easy to integrate tool in a proteomics laboratory workflow. And finally, I would like to thank all the members of the Functional Proteomics Laboratory of the National Center for Biotechnology in Madrid, Spain, for their support and help in this project, and especially to Fernando Corrales as head of the lab, and to Fatima Milano, Irene Hernandez, and Sergio Ciordia, who have been the people mainly involved in this project. I would like also to thank the support and collaboration of the members of Thermo, both in San Jose, California, and in Spain. Thank you very much for your attention.